Okay, so let me tell you about a story that happened to me. So this weekend, I was cooking actually yesterday. I was actually cooking, and I have my day plan usually every Sunday because I don't get a chance to cook throughout the week. So I eat a lot of fast food or a couple noodles or this because literally I have classes back to back to back to back to back. Letitia, since you're in two of my
Yes.
classes. Um, so since I am constantly on the run, I don't get a chance to cook. So when the weekend comes on Sunday, I try to cook at least one meal. Usually that's breakfast because I like breakfast. I'm big on breakfast. Something sweet, something savory. It's delicious, right? So and I'm currently seeing a lady and everything like that. And so she had to work a late night shift. So I was like, oh, I'll make her breakfast and take it over there and or brunch. And so I went out, made all my little mixes and all that stuff. And there's a dish that is kind of part of my culture. It's called mackerel. Mackerel is a fish that a lot of people don't like to eat. So in the South, poor people would basically have it because it'd always be around because nobody liked to eat it. And you'd make these like little mackerel cakes, kind of like crab cakes. So you use breadcrumbs or crackers or something like that mixed in with it. And so it goes a long way. Cut up an onion, but it's delicious. And you put it in syrup and eat it with biscuit and all that. But I made pancakes instead of having biscuits. So you can use the syrup and the pancake to kind of eat it. And I made hash browns and all this, right? So I'm in the process and I'm like using all my whole stove time. And all of a sudden I hear, pop! And I'm like, what the hell? What's going on? And and I look and I'm like, look at the stove and there's nothing wrong or anything like that. And then I'm like, okay. I hear pop again. And then I see this flash of light. And I'm like, what the hell? And then all of this heat, just expands and you kind of see the smoke and everything going off of it. And I'm like, what the hell? What the hell's going on? Right. So, I wasn't thinking, I've been running off of minimal sleep over the last couple of weeks. So my first instinct was to touch the burner. And so what happened when I touched the burner?
You burned yourself.
I burnt myself, right? So not only did I blow up the stove, I also burnt myself. So I have a nice little burn on my hand, right? And I was not quite thinking about things. So, but, fortunately enough, it's not too bad. It's just like, skin is all peeling now, but I touched it and then I moved it back, right? So we know that the nervous system is kind of important for that process. Without it, it won't work, right? And we know that this process, that reflective response, has to be delivered. We have to send stimuli to tell us what we need to do, right? Oh, hot, move your hand, right? So we have that sensory response that picks up the fact that it's hot. And then we have the reflective response that tells us, hey, or that motor response, hey, move your hand. Okay. So we're going to be talking about all of the kind of the components to that in more in terms of how that process happens. And then also we need to know a little bit of the people that are taking part in that. Okay. Or not the people, the components, the receptors, the nerves, the cells that are going to be involved. Okay. Okay, so the first thing that we're going to be talking about are the different types of sensory receptors. Okay. So the first thing that we kind of come in contact with in terms of making that initial response is we need to sit there and receive that sensory. We need something that's going to tell us, oh, it's hot. Oh, it's cold. Okay. So we're going to be actually classifying the receptors by the different cell types. Okay, so if you take a look at the figure above, so this is basically a neuron. So, and this neuron is going to be serving as our receptor. It's actually receiving the response initially. Okay, and if you kind of take a look at it, it has its, these guys, it's called a free end, because it has free ends, essentially. And then the other one is encapsulated, okay? Meaning that it's, it has basically a nerve ending. So free end nerves, they have the dendrites that are sitting there hanging out there. You're going to have your myeloid sheaths going along the line, just like all other neurons in the body. You're going to have, as you said, the axon going on between the myelin sheath. And you're going to have your cell body that's going to be located somewhere in that middle point, okay? But key is that you have both of your ends free, okay? So the free ends, basically, they're going to be the ones that are going to be embedded in your tissues. And they're going to be receiving, you know, a lot of your sensations. Okay? The nerve, the encapsulated nerve endings, so those guys are going to be the ones that are going to be associated with certain tissues that are going to be responsible for like enhancing, right? So like, for instance, your eye receptors, your photoreceptors in your eye. Oh, sorry, not those. So, for instance, the ones that are that embedded like your connective tissue. So that's become like your surface and your skin. So these guys are going to have to be responsible for like the location that of like your skin cells along that part, so they feel the touch, those type of responses. Where the free end nerves, like, those guys are responsible for picking up temperature and and heat and pain, and so these guys are going to be pain, and temp.
So you could say they both sense certain things.
Yes. So yeah, it's gonna be, but they both sense. They're both responsible for sensing. But this guy, because you have these guys that are encapsulated, right? So they're going to be a little more dampened, right? So these guys, since they're free, they're going to be able to sit there and pick up those very fine signals. Since these guys are more encapsulated, more dampened, so these guys are going to be kind of like on the skin. I mean, they feel touch, we feel touch, but it takes a little bit before we actually start feeling that pain, right? Because the skin is used for protection, so it has a little more durability, but it does feel the pain. These guys, both of these guys are going to be there, but this guy is going to be more of that dampening of that signal. So that's that encapsulated nerve ending is kind of more of a dampener. Does that make sense? So like, okay, and then we have what we call specialized receptors. So this is kind of like your eyes, you know, so the receptors in your eyes, the photoreceptors. Okay. So in the case of the free end and the the encapsulated nerve endings. So in the case of those two types, so these guys are going to be, they're going to happen bipolarly, right? So basically, sorry, unipolar. It's going to be happening at both ends, essentially, right? Where these guys are going to be happening on one end. So it's going to be dictated to a bipolar cell. So it's going to be delivering in a direction where, in this case, you're going to have both ends receiving the signal. So unipolar and bipolar. Oops. Okay. And you guys, like both of you guys have taken chemistry, so you guys are familiar with bipolar, meaning, you know, plus and a minus type of situation, right?
Mm-hmm.
So bipolar basically means that you're moving towards one to the other type cell, not having both characteristics. So, okay. So other ways that you classify receptors is we have okay, so we have the exterior receptor. So they're going to be located near the stimulus on the external, like the external part of the environment. The interior receptor, so that guy is going to be like your internal organs. And tissues. These guys, the proprial receptors. So these guys are going to be located next to moving parts. And that's going to be like your muscles and stuff. Okay. We also have what we call functional types of receptors. Oops. Okay, so we have, of course, chemoreceptors. Okay, so they're going to interpret chemical stimuli. So that's kind of like what's going to happen with your taste and things like that. So something that's sweet, it recognizes that it's sweet. Something that's bitter recognizes that it's bitter. Right? We have osmoreceptors. So basically pressure, osmotic pressure. Or let's see, I shouldn't say pressure. It'll be more like fluid balance. So like fluid concentration. So like, so you guys know like your cells themselves, like the concentration of your cells compared to the concentration of the outside. So if there's more solute on the outside, it's hypertonic. If there's more solute on the inside, it's hypotonic. And so your fluid basically moves across the membrane to sit there and balance out that concentration, right? So osmosis, the process of osmosis. So osmoreceptors basically recognize the concentration in those different areas. Okay, then we have Receptors. So these guys are, they're kind of like pain receptors. They're pain receptors that are kind of like adapted for like tissue damage. So like, let's say you have a, let's say I was running or something like that, and I ran into like a stick and it scraped my arm. And so the pain that's associated with that scrape, so it sends like a chemical signal and stuff like that. Stuff is getting in the pain that's associated with that. Okay. And then we have mechanical receptors. Okay, so these are they're gonna recognize like vibrations, pressure, anything that is more physical and more mechanical, you know, and so like pressure, vibrations. So those are going to be kind of like the stuff that you have in your ears. So those are going to be the receptors that are going to be used for like your hearing and stuff like that, so that we can hear the sound waves and stuff like that. So those pressures that are associated with that. And then we have temperature receptors. And that's kind of self-explanatory. Okay. So let's talk about your tongue. Okay, so this is basically, this is a diagram of your tongue. If you actually take a look at your tongue is covered by these little bumps, small bumps. They're called papillae, papillae, or papillae. So they're responsible for kind of recognizing the chemicals that you're going to get from your food, from, you know, salt, sugar, from your drinks, things like that. And so they have the receptors that are going to be located at that point. And so they're only going to be found in the small regions of your tongue, but different regions. So, and so like if you say we stick out our tongue, we're going to have different types of papillae that are going to be located. And I'm going to put the words up here because I don't know. Can you guys see that?
That's too tiny.
Yeah. I have to get better images. Okay. Okay. So okay, so there are going to be 4 types of papillae, and then they're going to be the fungiform, there's going to be the filiform, there's going to be the fuliform, and then there's going to be the circumvallate form, circumvallate. And these guys are all based upon appearance, how they look, right? So the fungus, the fungiform, basically the reason why they call it that, because it kind of looks like a fungus shape. And the picture doesn't do justice, it's hard to see in the book as well too, but it's just kind of like a mushroom-ish, the way the growth is. The forms are kind of like little spikes. So they're like, this guy's kind of like mushroomy-ish. So it's, you know, kind of mushroomy-ish. So they're kind of lifted, and then underneath, these guys are kind of spiked. And then these guys are just kind of all over that point. And so and then you have the circumvented, you know, again, it's based upon its shape. So they're kind of like a circular area that's kind of been kind of indented, you know, in a sense. So basically, the taste buds are going to be lined underneath these papillae. So they're going to be entrenched in there. And in your taste buds, you're going to have these specialized receptors. So these receptors, and they're receptor cells. So you have your gustatory cells, which are going to be located here. And let me spell that for you. And again, I apologize. I'll have to get better slides at some point. Okay. So again, these are going to be receptor cells. And they're going to be responsible for sending those signals, or transducing the stimuli, the taste stimuli. So you're going to come in, you're going to get your taste, the chemicals that are going to be sensed are going to be done at this point. And then these guys are going to be responsible for sending that signal to your brain, right? Through activating your sensory neurons. So it's going to activate it, it's going to send the signal, and then you're going to respond to that. Oh yeah, I like it, or oh, that is horrible, or oh, that's so bitter, right? So it's going to sit there and give you all of the different stimuli that you're going to get from the chemicals that you're getting from your food. Okay.
I remember one time I drank something hot. It usually happens once in a while. When I drink something hot, I think one of those little things flares up because it's just the one that's just it's more visible
Yeah.
on the sun, and it's just bothering it's just bothering me because it's just up there flared up until it, I guess, calms down, or it's tough.
No, yeah, I have the case. But usually, like when I drink something hot, I usually burn it. Like you burn the crap out of it. So yeah, so you're right. They're getting flared in that sense because they're irritated and they're trying to repair the damage that you've done. And so yeah, that definitely happens quite a bit. Okay, so let's see what else. Okay. And it's funny because there's different regions of your tongue that are going to be responsible for picking up different tastes as well. And so your tongue is going to kind of be, even though it has these pillae, each of those pillae have different regions, and they're kind of responsible for recognizing different things.
Yeah, I remember there was in school when I was in school for medical assistant. Carolina was divided in a certain area. So a certain area was like for sweetness, a certain area was like for sourness, and a certain area for bitterness, well, and so on and so on.
Yeah, exactly. No, that is exactly it, and it kind of tells you, it kind of helps you understand that, each of those, like, because you have these taste buds that are located all over the place, but these the papillae, they're causing that organization so you can distinguish between certain things without that interference. This is your job here. This is what you do. It's kind of like parts of factories that recognize, okay, so you do this part, and you do this part, and you do this part. So it recognizes different components. Okay. Let's see. Next, we have the olfactory system. So the olfactory system is basically our smell, our system of smell. Let's see, let me clear. I can't. I apologize for the bad text. So here we have the olfactory epithelium. And so that's going to basically contain your bipolar nerve sensory, your bipolar sensory neurons. And these neurons, they're going to basically have dendrites that are going to be extended on the apical surface of the epithelial into the mucous, the mucus lining. Okay, so the mucus lining of the nerve. So if you, let's see if I can get it. So if we take a look here, this is, so we take a look here. This is just basically that area blown up. And let me type this in so that you guys know what I'm talking about. So we have the olfactory. So we basically have our nerves, or the olfactory nerves that are going to be extending from this point all the way down to here. And so you can kind of see, here's your olfactory receptor. And it's going basically from this point all the way up to this point. Okay, so all of those networks. Here we have our dendrites that are going to be actually into the mucus lining itself. Okay, so if you think about it, that's the route to the external surface. So we have the mucus lining that's on the inside of our nose that kind of helps generate the snot and stuff like that. So if something happens, our nose flares up or something like that, it's able to recognize it. Or something is, they get a bad irritant, hot coffee, or we sniff in pollen, and we start sneezing as a result of try to get the pollen out. So those nerves are going to be sending those signals up to our brain, from the peripheral nervous system to the central nervous system, and so that we'll be able to sit there and respond. So those sensory nerves are really located through that process, and they send the dendrites that are in that process to recognize that. Okay, and so these neurons, right? So we have the olfactory bulb as well. So these axons are going to be projected through the cribriform plates of the modal bone and the synapses. The synapses of those nerves are going to kind of join to that olfactory bulb. So they're going to join up together. So if we take a look, so the olfactory bulb is here. And so if we're sniffing something, right, get a big whiff. Have you guys ever smelled vinegar?
Oh yeah.
Yes.
Okay.
It
So
smells
when you
awful.
so your first response is like when you take a big whiff of that, right? And you're not expecting it to be vinegar. It looks like water, right? You take a big whiff, and if you're, what was that?
It burns.
It burns, yeah, it somewhat burns the nose, right? Because those acids and stuff, they give that sour, or I should say, those positive ions that are sitting there in that vinegar are giving that sour bitterness to it, that kind of sour bitter smell, and it irritates, those receptors. It actually gets those receptors really started up, and so you respond immediately. You kind of switch up and pull away from the vinegar, right? Okay. So if you look here, this is basically just showing you the route, how the neuron is basically extended. So that's the modal bone. And then you can kind of see it going through the connective tissue and all of that, just to get to the point where it's actually embedded into the mucosa itself. So from the dendrite, we get the signal, it goes up to this process, and then it's going to connect with the join with the synapses of the olfactory bulbs. So you're going to have a synapsis point at that point where those two neurons are going to be joining together. Come on. Okay, so then we have hearing. So far we have had taste, our tongue. Now we've had our nose, smell, and now we have auditory. So we've kind of talked about, in terms of auditory, a lot of auditory is going to be dealing with those receptors that are going to be responsible for recognizing vibrations and pressure, right? Okay, So this is the makeup of our ear. And so you can see the articles, the ear canal, and actually this is a pretty good picture compared to the rest. We have our tympanic membrane and our tympanic cavity. And have you guys taken anatomy yet?
Yes.
Yes.
Yeah, okay. So you guys already know this, I don't have to talk about. So you should be familiar with all the different structures in the ear. The vestibule, the vestibulary nerve, the cochlary nerve, and then the cochlea. And so when we sit there, we look at the transmission of sound. So we get sound waves, and they're going to be kind of be delivered in terms of like pressure, right? So you're going to have either high or low pressure that is going to be kind of delivered to your ears. And so because of the vibrational modes. And so you're going to recognize those different wavelengths, and that's basically what sound is going to be made of, right? So that tympanic membrane basically is going to vibrate in response to the sound waves that you're getting. Those vibrations are going to be amplified across the oscillus, which is so it's located here, and that's, I don't know if you can see the spelling. Such bad. It's funny, I get such good images. They have such good images, but then when they blow up, they get really bad. Some of them. Okay, so then it's going to sit there, and it's going to, the vibrations are going to be against that setup and the fluid that's going to be in the vestibule. And so then you're going to go from those sound waves that are initially that were vibrating in air and across that membrane. Now you're going to be vibrating within the fluid itself, to a fluid, and that fluid is going to sit there, and it's going to cause pressure bands in the membrane. It's going to cause the pressure to kind of bend the membrane a little bit, okay, of your cochlary duct. And then, so then that vibration is going to kind of give a frequency, and then the hair cells that are in there with that membrane are also going to vibrate. Okay, so in that sense, that signal is just going to be kind of conducted through that process. So the initial sound waves going in with being just that pressure, then the tympanic membrane is going to vibrate in response to those sound waves. Those vibrations are then going to be amplified by the across the osculus. The osculus is going to cause those vibrations to sit there to cause waves in the fluid, and then those guys are going to sit there and just build up pressure bends along that membrane. So it's going to cause this membrane to sit there and bend. And then that's going to give like frequencies that are going to be kind of picked up by the hairs in the basilar membrane. And those hairs are going to be vibrating. Okay, so where are we? Okay. So we sit there. This is a cross section of the cochlea, and so there are going to be three major regions of the cochleae. So the three major spots that are highlighted. So we have the scala vestibuli. We have we have the cochlear duct, and we have the scala tympani. Okay. And these guys kind of lie on both sides of the cochlear duct. So this guy is going to be responsible for the transduction of your waves. And it's going to be transducing the waves that are going to be picked up by this guy and by this guy. So these guys are going to be picking those up, and then it's going to be going in here, and then that's going to be transduced. Okay, and so this guy is going to have those nerve endings that are going to sit there and send that signal. Okay. Okay, and so there's the bacillary membrane that kind of lies in there, and it's going to take those wave frequencies, and then transmit that to your nerves. And so in your cochlear branch. Does that make sense?
Yeah.
Yes.
Okay, so come on. Okay, so this kind of explains those hair cells that are found in our ears and everything, right?
Mm-hmm.
So the hair cells are going to be basically, they're going to be mechanical receptors, and they're so these guys are going to be these guys are going to be able to sit there and bend, and their bending, right, will sit there and send signals. Okay, and so they're going to basically cause a membrane to move. So these guys, by these guys bending, they're going to be tethered to via the stereocilia. So, and this is going to cause this guy to move. Okay. The pectoral membrane, and then that movement there, right? So that movement is going to be it's going to cause your channels to open up, your ion channels to open up, and so you're going to get that depolarization, and then that electron potential to start sending it down that pathway. So this here is just a cross section of the cochlea and the organ of Corti. and I should say, let me go back. So these hairs are going to be located in the organ of Corti. And so this is just that cross section, just kind of showing you where the location's at in the inner ear. Okay, so those frequencies are going to sit there and be coded in the colea. So if we sit there and we take a look at it, the colea is going to be able to sit there and distinguish between our high frequencies, our middle frequencies, and our low frequencies. And then so the way that it distinguishes between that is going to be the relative length of the fibers of in that membrane and in the basilar membrane. Okay, so the hairs are going to be activated on the only by the high frequencies, and then the hairs that are at the base of the colea are going to be able to be activated by the ones that are in the high frequencies, where the apex of the colea is going to be activated only by the ones that are in the low frequencies. So
the hair kind of determines the wavelength of it, of the sound?
Yes, and it's all based upon, so it's based upon the length of your fibers, so the relative lengths of your fibers. So the earlier one, right? So at that point, you're going to recognize just those high wavelengths. So basically the membrane is going to be moving, and as a result of that membrane moving, it's going to sit there and translate that to kind of frequency. Those hairs are going to respond to the different frequencies. So it goes through and says, okay, this region is going to be recognized but the high frequencies. This region is going to recognize the middle frequencies, and this region is going to recognize the low frequencies. But the distinction between them is their location and then also the length of the hairs, fibers themselves.
So the one that would determine the little high pitches, like the ones that you can barely hear, would be in the far end, or?
Yes, so those are the ones that are going to be the first recognized. Yeah. So your high frequencies are going to be located at the far end, and then at the apex, you're going to get your very low frequencies. Oops. Okay. So, and the key again is that it's going to be, there's going to be relative, so the relative length of the fibers is going to dictate which frequency is going to be recognized and where it's going to be recognized within that region. Okay.
It's amazing. I think with the ear, it's amazing how you can lose the hearing and can't gain it back.
Yeah.
How come you're not able to recover your hearing if you lose from either your left or right?
Part of it is the cells that are being replaced. It's like cells there, the rate of replace occurs so slowly that you don't have enough time to sit there and do it. And then also, you have all of the different structures that are responsible for that transmission, that signal transductions, those membranes and things like that. The replacement of that is very difficult if those cells aren't being replaced during times. And so if you think about it, those cells, they grow fairly slowly in the first place. And then as we age, so they're going to be growing even slower or even worse. And circumstances. So as we age, we start to lose our memory as a result of that, because the cells, the replacement of those cells are very slow. And then also, when we cause damage to our cells, we also cause enough damage that they're not being replaced in a frequently enough manner to sit there and repair the damage that, like in the case of a wound or something like that.
Okay.
Does that make sense?
Yeah.
Yeah. So like, for instance, so these guys here, along here, you have all of these different little hair structures, right? And again, what I should say, we're talking about the length of that canal. Then we're also talking about the length, you know, of the little hair fibers. They're not too far from each other, but there's going to be a slight difference between them. And that slight difference is what's going to sit there and dictate that frequency. So we have hair cells in there, but like in the case of our hair out here, we lose that hair, we continue to grow that hair. Where in those inner canals, they're limited in terms of how fast they can grow, those cells can grow. And then we're also limited by the frequency that they can grow, because, you know, it's not, they don't have a readily outlet to sit there and get out there. So we tend to make, you know, our earwax and stuff like that, that will have some of those hair particles and stuff in that process being disposed of, but there's a limited space and there's a limited room for growth and regeneration. And because of that, those cells are very specialized and they grow very, very slowly. And then, as I said, as we age, that control of that growth diminish, you know, in a sense. Let's see, linear. Okay, so if we sit there, we think about equilibrium. Have you guys heard of equilibrium or your balance?
Yes.
Okay, and you know that like your balance is kind of controlled by your inner ear, right? You've heard of that before, probably when you're in anatomy.
Yes.
Jesse, you're here. You okay?
Yeah,
Yeah.
just had a few issues, and I don't have phone service currently.
Oh,
So,
okay.
yeah, I'm good
Okay.
though.
Let's see. Okay, so part of this this inner ear equilibrium, it's going to be like considered our sense of balance. So this is going to act very similar to those mechanoreceptors receptors with the hair cells in the with the stereocilia. So in this case, they have like this individual whose like head is upright, and that kind of represents, like, as I said, our balance, right? So these hair follicles are going to sit there that are going to be they're going to be the see, how can I say this? So this here is the tissue, and those hair follicles are going to be embedded in that tissue, and they're going to be connected to nerves internally. And its job is to basically, it's going to be sensing that it's going to be going through the otholytic membrane, and which is basically going to be a layer of calcium. So this here is just like a layer of calcium and calcium carbonate. And so that layer there is going to be it's going to be tilted based upon our head movement. So like if we're even eyes leveled, it should be even. If we're tilted, then gravity is going to be pushing it downward. And then, so then as our head tilts forward, it's going to kind of affect that concept of balance, right? So in the case of, let's see, can you guys do me a favor? So start with your head looking forward, and then just kind of, when you like bang your head, you know, just kind of like, do you feel like a sense of pressure that is kind of stemming from your inner ear?
No, it's mostly from the back.
So you're saying it from the back of your ear or the
back of my head.
okay, so then that has to do with your brain moving a little bit. But like,
okay.
Like when you, I guess, don't do it hard, just kind of push your head forward and down, like, but kind of firmly, like. And do you feel a sensation that's happening, like a pressure that fills up, like at this point?
Pressure.
Yeah, in your ear, right?
Yes.
Okay, so all it is that membrane, because of gravity's pushing on those hair motions, you feel that sense of pressure, right? And that discomfort. So it's like a little pressure from just that momentum of moving forward. Does that make sense?
Yes.
Okay, now move your head like this. So do you feel any sensations around your ears when you?
Yeah, like a little whooshy.
A whooshy, right? Okay.
Yeah.
So when we're moving, we're rotating, right? We're going to be using the semicircular canal in that case. And so there's going to be hair follicles that are going to be going through there as well. And so when that moves, right, the cupula, when that moves, it's going to cause those hairs to be pushed, right? And so when you're moving back and forth, you can kind of feel that pressure that builds up. That's from that movement of the copula bending the hair follicles there. Okay. Okay. So. So if you guys, I'm gonna put up a whiteboard real quick, and then I'm gonna add something here. How are we doing on time? I have 10 minutes, huh? Okay, so what I'd like you guys to do is kind of be familiar with the table that's on it's on page 614. Table 14.1. And it's so these are the mechanical receptors of somatosense. So be familiar with that table and kind of the key components. And remember that, again, a lot of it deals with those hair follicles being moved that are being embedded in different tissues. And so when those tissues are mobile or shifting due to gravity or during motion, then it's causing those hair follicles to send that signal. Okay, let's see. I didn't get as far as I wanted to. Okay, the next thing we're going to talk about is the eyes in the orbit. So, in the case of the eyeball itself, so the eye is going to be basically located in the orbit. It's going to be surrounded by tissues, and those tissues are going to be responsible for protecting it, and it's also going to be responsible for the eye's actual function. Excuse me. So if you actually also take a look, that eye has bone that's going to be located, the cranial bone. Cranial bone. I'm showing you the wrong slide here. Okay, so the cranial bone. And so you're going to actually have nerve endings that are going to sit there and that are going to be going through there. And that's going to kind of be important here. Oops. Okay, so we're going to skip most of the structure of the eye, but we're going to go to the photoreceptors itself. So if we take a look at the photoreceptors in the eye, excuse me. So these receptors are going to have segments that are contain the organelles and the nucleus. Then we're going to have the outer segments that are going to contain the opsin molecules, which again, I apologize for the bad images. So you're going to have your rods, which are located here in green. So these guys are going to be your rods. So these guys, and if you take a look, where the nuclei are going to be located at. And again, these guys are an example of bipolar, these bipolar receptor types. And then we have your bipolar cells. And then down here you have your ganglion cells. Okay. The gray, those, I should probably put them in different colors. These guys are your cones. And this here, they're really hard to see, but this here represents right outside of the nucleus. you usually have your Golgi apparatus. And so it's part of that secretory pathway. And so your photosensing molecules are going to be secreted secreted outside of the cell or towards, so it's going to be delivered outside towards the end parts of the cell. So it's going to be important that these guys be oriented in the right way so that you have a pathway so that all of the things are being delivered in the appropriate manner. Okay, so so you have polarized secretion in this manner. So where in other cases you don't necessarily have, it's not necessarily polarized or delivered from a apical spot to a basal spot or a directional type of secretion. So you guys in chapter, I think it was chapter 3, when we were talking about the cells, and we talked about all the different organelles, and we talked about vesicles that were going from the ER to the Golgi to the vacuole to the cell membrane, right? So in certain cells, that is happening in no organized fashion. You know, you have the nucleus in the center and it's just going out all around the cells. In these cells, this orientation is essential to have a direction that it's going in, because you need those photosensing molecules at various areas, and you don't want them to be going internally, because internally, these guys are going to be connecting with other secretory vesicles that are responsible for signaling, right? Signaling and changing the that action potential, so depolarizing the thing. So those synaptic connections, right? Does that make sense at all?
A little bit.
Okay, so okay, so in the second figure, right, this is just basically tissue of the retina showing showing you the dense layer of nuclei. So the nuclei are going to be kind of oriented, kind of close together. So they're going to be uniformly oriented, so that you can kind of see where the nuclei of the rods and cones are at. You take a look at it, your rods, long, lankier type of structures, and then your cones, more broad, shorter, stouter, type structures. Tapered, as they say in the book. Okay. And so we have light entering our system, and then when light enters, then that's going to be delivered to those bipolar cells, that's going to be delivered to the conal cells, and then here, this is where we're going to have our pigment, and so that's where you're going to have your rhodopsin pigment and stuff here, in that in the epithelium, the pigment epithelium. So this the term isomer. Isomer basically means same chemical formula, but different chemical structure. So when we have a molecule that has the same chemical formula, it has the same number of carbons, hydrogens, and oxygens, but it has a different structure. That structure can affect things differently. So in the case of retinal, it has, we have retinal 11, and then we also have what they call all trans retinal. In one case, if you take a look at the pathway, this here is basically clear. So this here is rhodopsin. These are rhodopsin molecules. These circles, these are your disc that are in your rods. And so what happens is that your rhodopsin molecules are going to be used for kind of picking up that light. So the photon, the rhodopsin molecule, so rhodopsin is a component of opsin, and your retina, one of your isomers of retinal. The photon gets captured, or the energy from the photon gets captured in the rhodopsin. It causes a change in the retinal. So then the retinal becomes all trans retinal, or sorry, 11 trans retinal. Then it gets what they refer to as bleaching. It goes from 11 trans retinal, using that energy and using energy from ATP, it goes from 11 trans retinal to 11 cis retinal. And you break up that retinal and the adopsin, and then the adopsin gets recycled. So then adopsin goes back up here, and it interacts with it'll interact with the 11 cis retinal to form rhodopsin. So okay, this should be 11 trans. In your book, it says all cis, all trans. It should really say 11 trans. Okay. The best way of understanding trans and cis, trans is across and cis is the same side. So if we had a double bond and we had a you guys remember organic. So this here would be cis. This would be trans, where they're across from each other. Cis meaning the same size, trans meaning across. Transatlantic, across the Atlantic. Okay? Are you guys there?
Yes.
Okay. Okay, so your lab today is going to be the physiology lab I didn't put up yet. All right. It's going to be a Labster lab, and I'll be putting it up. And then on Wednesday, we will actually be using our fish again for a stimuli experiment. We'll be doing the last part of our fish experiments. It'll be working with the alcohol and the caffeine. Actually, we should probably do them separately. So we're going to do the alcohol this week, and then we'll do the caffeine next.
Okay.
Okay?
Mhm.
Okay, good. Okay, actually, that isn't true. We have alcohol, caffeine. We have 2 this week. We'll do the light and the alcohol this week, and then the caffeine next week. Sorry about that.
For the page that you gave us, you just want us to look over the table?
Yes, look over it, be familiar with it, because on the exam, you're going to have something to deal with that table.
Oh,
Not on this exam, but the exam after, in 3 weeks from now. Which will
So
probably be your
our
last exam. Your last exam
So-
before your final. What was that?
so our practice and our second exam is due on the same day on Friday.
Your say that again, please.
If our practice and our second exam is due on the same day, which is Friday, this Friday.
Yeah. So I did that because a lot of times I have I'll give you guys a practice and I'll have it 2 days before, and then I always have those students that they wait till the very last day to do the practice, and then they'll contact me and say, "Can you open it up?" And I just have them both due on the same day.
Oh, okay.
Okay, but you can do it. The practice, you can do it as many times as you want. You know. Okay.
All righty.
Okay. I will see you guys on Wednesday. And
Okay.
you have a good day.